Hello and welcome. My name is Emily Priya, and I will be your lecturer today on ATP pathways. There are 4 key steps in glucose metabolism. Now, this can happen in an anaerobic, meaning without oxygen, or an aerobic setting, which means with oxygen. For our intents and purposes, we're going to speak about it happening in an oxygen-rich environment. The first step is called glycolysis, which is the separation of sugar into pyruvate. The second step is going to be pyruvate oxidation, where we take that pyruvate formed in glycolysis and oxidize it. Move it into the third step, which is the citric acid cycle, or otherwise known as the Krebs cycle, and into our fourth step, which is oxidative phosphorylation. In this slide, we're going to be talking about the first 5 steps of glycolysis. Again, we talked about glycolysis being able to happen in either an aerobic with oxygen or anaerobic without oxygen environment. When it is aerobic, it is a little bit slower of a process, but it does produce a substantial amount more ATP than in an anaerobic setting without oxygen, though that is a much quicker form of ATP production when it comes to glycolysis. So step 1 of glycolysis, the glucose molecule, which is a 6-carbon sugar, moves its way through a glut transport. Now that glut transport is bidirectional, which means that it can move in or out of the cell. In order to keep it in the cell, our body has to use the phosphate from 1 ATP, or adenine triphosphate, and turn that into ADP, adenine diphosphate, with the help of hexokinase. That hexokinase takes off one of those phosphates from ATP and tags it onto the end of the glucose molecule, making it the G6P molecule, which is a glucose 6-phosphate. In step number 2, remember in step number 1, we have moved that phosphate from ATP tagged it onto the end of the glucose molecule and created G6P, glucose 6-phosphate. In step number 2, we swap atoms around, and that G6P turns into a fructose 6-phosphate. They're fairly similar, just things are a little swapped around. This primes the further metabolism of the glucose molecule. Now we move on to step number 3. Step number 3, again, costs us 1 ATP, and I bet you can guess why. Again, hexokinase is going to swoop in, steal one of those phosphates from ATP, turn that into ADP, and tag that on the end of the fructose 6-phosphate, so that we then have 1,6 bisphosphate, which is a fructose bisphosphate. The reason we say bis instead of biphosphate is because the phosphates do have some space in between them. In step number 4, fructose 1,6-bisphosphate is cleaved through the help of catalase, and this turns into a D-glyceraldehyde 3-phosphate. And there again are 2 of them, and it also has a product of dihydroxyacetone phosphate. Now, we cannot use the dihydroxyacetone phosphate, so in the next step, step number 5, we are going to turn that dihydroxyacetone phosphate into D-glyceraldehyde 3-phosphate with the help of triophosphate isomerase. This turns it into 2 units of D-glyceraldehyde 3-phosphate. So now we're going to move on to steps 6 through 10 of glycolysis. In step 6, we have our D-glyceraldehyde 3-phosphate, and that is then catalyzed by glyceraldehyde 3-phosphate dehydrogenase, which makes a product called D1,3-bisphosphoroglycerate. This happens twice, remember that, and in the process, 2 molecules of NAD are then converted into NADH. Remember, we just used dehydrogenase. So where'd that hydrogen go? It got added to the NAD. In step 7, we gain 2 ATP. Now, those 2 ATP happen because our 2 D1, 3 bisphosphoglycerates repel each other, right? And that repellent allows us to lose a phosphate. And now this is sometimes coined as the Big Bang of glycolysis because a lot of energy is created during this time period. It allows the phosphoglycerate kinase, the enzyme, to phosphorylate 2 more molecules of ADP to create ATP. So we move on to step 8. In step 8, our 3-phosphoglycerate is transformed into 2-phosphoglycerate. Oxygen is also released during this time period, and it binds to the hemoglobin. In step 9, we're back to gaining. We gain 2 more ATP. Now, we gain that by having our 2-phosphoglycerate converted into an enol by enolase, and then it turns that into phosphenolpyruvate by removing the water. And now where do you think those 2 phosphates went? Again, they went over to ADP to be converted into ATP. Finally, our last step, step 10. We have our phosphenolpyruvate, which is converted to pyruvate, which was our main goal at the end of this, by pyruvate kinase. The phosphate from that phosphenolpyruvate, or PEP, is added to ADP to create ATP. All right. And here you'll see the chemical chart of how glycolysis happens. Remember, we've got our first step in which glucose enters the cell. The next step where hexokinase is adding phosphate to create ATP to make our ATP, add that phosphate to trap glucose into the cell, move into step 2, step 3, step 4, all of which at the end our goal is to create pyruvate so we can move on to the next step of glucose metabolism, which is pyruvate oxidation. So now we have pyruvate oxidation. In pyruvate oxidation, we can have this happen in an anaerobic or aerobic environment. In an aerobic condition, pyruvate is converted into acetyl-CoA, which is what moves on to the next step in the citric acid cycle or Krebs cycle. So that moves into the mitochondrial matrix, which is the innermost space of the mitochondria. Before it was in the outer space of the mitochondria. It's transformed into a 2-carbon compound by removing carbon dioxide. And this removal then produces 2 NADH, right? Because we've removed hydrogens. Coenzyme A, or CoA, which is made from B vitamin B5, then reacts with this molecule, and it results in acetyl-CoA, which is what, again, moves on to the citric cycle acid or the Krebs cycle. Now, this is when conditions are really good. We have oxygen present. When oxygen's not present, when we're in an anaerobic environment, things change a little bit. Now, in that anaerobic environment, pyruvate is instead converted into lactate. Now, without that oxygen, lactate then enters into a process of fermentation. This fermentation is very similar to that of yogurt or any other fermentation process where oxygen is not present. It converts the 3-carbon pyruvate into a 3-carbon lactate. While regenerating NAD, because there isn't quite enough of it, since it's in limited supply, it must be regenerated, and it allows ATP production to continue. So where does this lactate go, the 3-carbon lactate? It has a couple of different places that it can go. It can move into the liver to be possibly turned into glucose. It can also move into ATP creation, or it can be stored. It's really up to what the body needs at that given time. Down below, you'll see exactly how pyruvate is turned into acyl CoA through the help of acyl coenzyme A. And on the bottom right-hand corner, you're going to see how lactate dehydrogenase turns pyruvate into lactate. Now we're moving on to the citric acid cycle, or the Krebs cycle. The Krebs cycle begins by combining the acetyl-CoA that was created in the previous step with a 4-carbon carrier molecule, which then makes a 6-carbon molecule of citric acid. This citric acid moves through the Krebs cycle in order to gain energy. Now, it's important to note that for each acetyl-CoA that enters into the cycle, 3 molecules of NAD are reduced to NADH. There's 1 molecule of FAD, which is reduced to FADH. So if you need to know what NAD stands for, that's nicodiamide adenine dinucleotide. And FAD is flavin adenine dinucleotide. So you have these molecules of NAD that is reduced to NADH, and then 1 molecule of FAD is reduced to FADH, as well as the production of 1 ATP during this cycle. So total energy released in this cycle, 2 ATP, since everything happens twice, 6 NADH, because 3 of them are created for each of the acetyl-CoA, and 1 FAD, so that ends up being 2 FADH. Okay, so now we have oxidative phosphorylation. Oxidative phosphorylation is the process through which electrons flow down the electron transport chain. So the NADH and the FADH2 that were created in the citric acid cycle then start to deposit their electrons into the electron transport chain. And their protons ultimately become 2H2O. The NAD and the FAD then, once they've deposited their hydrogens, go back into their empty state of NAD and FAD. As the electrons move down the chain, energy is released through each step and is used to pump the protons out of the matrix, forming a gradient. So you can imagine the electron transport chain like a water ladder. So as it moves down, it moves from high energy to low energy, eventually losing its electrons as it moves down, with the ultimate product being water. This energy is harnessed by ATP synthase and allows the protons to flow back into the intermediate space to the matrix. And this energy is then coupled to synthesize higher energy ATP by phosphorylating ADP, which is lower energy, and with its inorganic phosphate into ATP. Finally, at the end of this, oxygen then accepts the remaining electrons and protons and forms water. So now we have fatty acid oxidation. Fatty acids go through a process to be oxidated. Since they're relatively inert, they need to be activated. Now, fatty acids come from the digestion of triglycerides and other lipids, and they're broken down through a series of sequential reactions that produce ATP. Some of these reactions do require oxygen, as well as NAD, so nicotide, adenine dinucleotide, and flavin adenine dinucleotide, or FAD. Unlike glucose, though, the breakdown of fatty acids produces a much higher yield of ATP. It is a slower process, but the yield is substantially higher than glucose metabolism. The enzymes that participate in the fatty acid catabolism are located in the mitochondria. And this is extremely important for the efficiency of energy production, as well as production of the fatty acids into ATP. Fatty acid oxidation is initiated in the outer mitochondrial membrane. So unlike glucose molecules, fatty acids, like I said, are relatively inert, and they must be activated through a conversion of that fatty acid into, or palmitic acid, into fatty acid coenzyme A. Okay? And so for each of those fatty acids activated, one molecule of acyl-CoA and one ATP are used to help the acyl-CoA synthase. Continuing on with fatty acid oxidation, fatty acyl-CoA then diffuses into the inner mitochondrial membrane, and it combines with carnitine. This is catalyzed by carnitine acyltransferase, which makes a lot of sense since we're working with carnitine and acyl-CoA, fatty acyl-CoA. It's just a carnitine acyltransferase. The acylcarnitine, which it's converted into, is a derivative, and it's transported into the mitochondrial matrix and then converted back to its original form of acyl-CoA. So inside that mitochondrial matrix, further oxidation is performed. These 4 steps will repeat continually until the beta oxidation, they're known as beta oxidation, and they'll repeat continually because the beta carbon encounters a progressive removal of 2 carbon atoms from the carboxyl end. So this will repeat until all of the carbon atoms have been removed from the fatty acid, and we have what we need to move into the citric acid cycle. The steps of fatty acid oxidation. So we have step 1, where the formation of an alkene in an oxidative reaction is catalyzed by acyl-CoA dehydrogenase. And the coenzyme FAD then takes 2 of those hydrogen atoms from acyl-CoA in order to form FADH2. In step 2, the transalkene is hydrated in a reaction that's catalyzed by acyl-CoA hydratase. So unlike in glucose metabolism, where we removed water, in a fatty acid oxidation, we do hydrate that in order to continue along in the process. In step 3, which the secondary alcohol is oxidized by a ketone, beta hydroxyl-CoA dehydrogenase, with NAD as the oxidizing agent. Then reoxidation of NADH to NAD then supplies 2.5 to 3 ATP in the process. In step 4, the beta ketoacyl-CoA is then cleaved by a molecule of coenzyme A, which then creates acyl-CoA and fatty acyl-CoA that have been shortened by 2 carbon atoms as the product. And like I said in the previous slide, these 4 steps then repeat themselves until all of the fatty acyl-CoA is degraded. So all of the acyl-CoA that's obtained may enter the citric acid cycle, or the Krebs cycle, and be used for the formation of either ketone bodies, or serve as a starting material for the synthesis of fatty acids, or move into the liver to create glucose. And here you'll see a chart that breaks down each of the steps of fatty acid oxidation as we move into where we end up with acyl-CoA as the end result. So which one is a better metabolism pathway? Which one? Should we say glucose is better? Should we say fatty acid oxidation is better? Well, if we look at the numbers, fatty acid oxidation does produce, like I said, a substantial amount more of ATP than glucose does. The only problem is it's a much slower process. So if we needed that for, say, a sprint or a heavy lift or something that required quick uptake of ATP, it wouldn't necessarily be the best choice. One molecule of glucose makes 38 ATP molecules. Now, if we were to multiply that by 7.4 kcals per ATP mole, we're going to find that one molecule of glucose is equal to 281.2 kcals. For fatty acid oxidation, well, we're going to get, from that palmitic acid, we're going to get a grand total of 129 ATP moles. That is a huge difference, almost 100 more ATP moles from fatty acid oxidation. If we're to multiply that 129 by 7.4 kcals per mole of ATP, we're going to get a grand total of 954.6 kcals for just one molecule of palmitic acid. So why is it that we don't just use fatty acid oxidation for the vast majority of our ATP needs? While both glucose metabolism and fatty acid metabolism pathways play huge roles in enabling us to sustain movement and engage in exercise, they are not created equal. These systems have to be interconnected in order to supply that continuous amount of ATP that our body needs for many processes, not just exercise. But when it comes to exercise, we use a combination of both glucose metabolism and fatty acid metabolism because our bodies are extremely efficient. So glucose metabolism is used during high intensity exercise, and it typically serves as the main energy pathway when you're doing something that requires you to have a quick speed or a quick amount of power. But the drawback is it doesn't have very much ATP that's available. And it does take quite a bit since we are in a anaerobic environment and anaerobic in a condition when this is the case, because the higher the intensity of exercise, the lower the amount of oxygen available in the environment. So what's the other drawback of that quick ATP generation is lactate. And lactate is very acidic. So our body has to go through much more to try to transfer that lactate through fermentation process like we talked about to create more ATP. And we don't want our bodies to become too acidic. If you ever feel that burn in your muscles when you're exercising, that's lactate. So remember, as your intensity of exercise goes up, the amount of oxygen in your system that's available goes down. So it can only produce a limited supply of rapid ATP. And that's why when it comes to low or moderate intensity activities, our body tends to rely on the fatty acid metabolism. Now, the reason this is because those prolonged endurance activities really can benefit from that huge gain in ATP. Remember, we talked about 38 ATP in glucose metabolism versus 129 ATP in one glucose of palmitic acid, one molecule of palmitic acid. So that metabolism process for fatty acid it requires oxygen, and it's definitely more efficient, but that's why we don't use it in a high intensity situation. It operates at a much slower pace, and when it comes to producing ATP, it makes it less suitable for high-intensity activity to use fatty acid oxidation or fatty acid metabolism in place of that glucose metabolism. The other thing is our brains tend to use almost primarily glucose metabolism systems. And the reason for that is that there are GLUT channels, GLUT transports in not only the neurons, but the blood-brain barrier. I mean, there's all different types of GLUT transports, but they are located in the brain and in neurons themselves. Those GLUT transports are also located in the muscle, the kidneys, the pancreas, and the liver. So there's a lot of opportunity for our body to use glucose metabolism when needed. So that was my presentation, and I really hope that you kind of gained a good idea of what metabolism for glucose looks like, what metabolism for fatty acids or palmitic acid looks like, and how those processes do tend to happen in the body. Though they are very complicated, they could be and can be understood if you take the time. Thank you very much.